In this lecture, we're going to talk about what are chromosomes and how does the process of cell division work, and then we are going to focus on life cycles, especially plant life cycles. We'll first address why have sexual life cycles at all rather than just engage in asexual reproduction or reproduction without sex. We're then going to quickly define what chromosomes are, and we are going to go over the basics of two forms of cell division. These are mitosis, which is the division that maintains chromosome number, and meiosis, which is a division that reduces chromosome number. We will then talk about the life cycles of early arising groups of plants, and we are actually going to focus on the ferns. So plants have sexual life cycles. This is important because it allows for genetically variable offspring. Individuals, at least most individuals, we will see have two copies of each trait. The copies for each trait are called alleles. With the sexual life cycle, each parent gets to pass down one version of each trait, or one of their two alleles. So this means that if the parent has one better version and one worse version, for example, maybe one version that makes a protein correctly and a second version that maybe makes the protein a little bit incorrectly, then since it passes down only one of the two versions to each offspring, some of its offspring are likely to get the better copy. Of course, the flip side is that some offspring will get the worse one, but at least some of its offspring will be well equipped with the good one. In sexual life cycles, two parents contribute hereditary information, and this is in the form of DNA, to their offspring. Since two parents are contributing DNA, that means the offspring are going to have new combinations of traits. Those offspring will also have two copies or two alleles for each trait. One will come from their mother, the other will come from their father. And at least some of these new genetic combinations of traits are likely to be better than the parents' combinations of traits. So at least some of the offspring are going to have a better chance of survival and reproduction. And we can refer to survival and reproduction as generating an individual's fitness. So by having genetically variable offspring, then it is likely that at least some offspring from a parent are going to have higher fitness than other offspring and even the parent itself. This is going to then allow for the possibility of improvements to occur over time. In a life cycle without sexual reproduction, none of this would be possible. In that case, all of the offspring would have the same two copies of each trait that their parent had, and each offspring would have only one parent. This would mean that if the parent had a bad version of a trait, a bad allele, then all of the offspring would inherit that bad allele, and there would be no way to get rid of it. So that wouldn't allow this process of getting rid of bad alleles and making new combinations of the better alleles, and there would not be genetic improvement over time. So when we talk about sexual life cycles, we are going to talk about a process that reduces the number of chromosomes in each cell by half, and we'll see that process is meiosis. Before we talk about that, let's talk about why it's necessary. And we're going to start with an example of what would happen if we had sexual reproduction without this stage where the amount of DNA gets reduced by half. So remember, in sexual life cycles, hereditary material, or DNA, from the two parents combines in the offspring. So on this diagram, let's see what would happen without a method for reduction. Here we have one parent, and it has a pair of chromosomes. Remember that each parent has two versions of each trait, so they have two matching chromosomes. For simplicity, we're going to look at cells with only one pair of chromosomes each, but there could be 2 or 3 or many pairs of chromosomes in each cell. So here's one parent, maybe it's the mom. Here's another parent, maybe it's the dad. They each have their pair of chromosomes. If there was not a reduction stage before sexual reproduction, then when they both contributed DNA to their offspring would have 4 chromosomes rather than the 2 the parents had. Then if this offspring reproduced with another individual that also had 2 sets of chromosomes, then the offspring from that event would have 4 pairs of chromosomes. And you can imagine this offspring, if it reproduced with another individual with 4 sets, would lead to an offspring with 8 sets, and every generation the number of sets of chromosomes would double. You can imagine that this would not be sustainable. Pretty quickly the entire cell would be filled with nothing but DNA, and each cell would have many more copies of each trait than it needed to do the things that the cell needs to do. So to avoid this situation, we need a way of reducing the amount of DNA prior to the stage where both parents contribute DNA to the offspring. So here's a diagram that shows what actually happens when we reduce the amount of DNA by half before the DNA recombines. And here again we have one parent on the left, another one on the right, and in this diagram we see that each parent reduces the amount of DNA it contributes by half and then contributes one of its 2 chromosomes to the offspring. This way the parents each have 1 pair of chromosomes and the offspring also has 1 pair of chromosomes. Then if this offspring reproduces with another individual that also has 1 pair of chromosomes, again we have reduction and then the fertilization event, then their offspring still has 1 set. And you can see that no matter how many generations occur, there is no increase or decrease in the amount of chromosomes per cell. So we've mentioned the word chromosomes, and this is probably an idea you're familiar with, but let's just make sure. A chromosome is a long continuous segment of DNA, and that's pictured with this line here. Along this section of DNA, there are many smaller sections called genes that each have information for one particular trait. So just for example, one segment of DNA might code for the shape of a thorn, whether it's curved or straight, for example. Another section of DNA might affect flower color, whether a flower is red or yellow, for example, and the third section might affect the size of a seed, whether the seed is large or small. So eukaryotes are individuals that have complicated cells that have internal membranes in the nucleus, and we've talked about that in a previous lecture. Eukaryotes have sets of chromosomes in each cell. They don't just have 1 chromosome per cell, or even 1 pair, but they typically have 2 or many pairs of chromosomes in each cell. Each different chromosome type has different genes on it. So there might be another pair of chromosomes that have genes for the shape of the root, or the shape of the leaf, or the size of the flower, for example. In most animal and most plant cells, there are 2 copies of each chromosome. And in cells with 2 copies of each chromosome, this is called diploid. The root di means 2, and so this is 2 ploids or 2 chromosomes. And having 2 copies is important because if the individual has a bad version of a trait, like maybe a gene for thorn shape that's not functional, then as long as the cell is diploid, then on the other chromosome there's a good chance that the other version of that gene or that other allele is going to be functional. So by having 2, this gives the cell redundancy in case one copy of the gene is flawed. In cells that undergo sexual reproduction, they need to have had a reduction in the number of copies of chromosomes from 2 to 1. So these cells are going to be called haploid cells. The prefix haplo means single. So these are cells with only one set of chromosomes per cell. The process of combining two cells together and allowing their chromosomes to come together in the nucleus of the daughter cell is called fertilization. So fertilization combines chromosomes from haploid cells to form a diploid cell. And you can see that in the diagram here. We have a chromosome from one parent, a chromosome from a second parent. They come together, and these two haploid cells then, after fertilization, form a diploid cell. This diploid cell is going to be called a zygote. The zygote is the first diploid cell formed as a result of fertilization. So we're now going to talk about two different forms of cell division. The first form is called mitosis, and this is cell division that maintains the chromosome number. The other process that cells can go through is called meiosis. We will talk about that next. In mitosis, the chromosome number in the original cells and the daughter cells stays the same. So let's look at this on a diagram. We'll start on the left-hand side, and let's look at the process of mitosis in a diploid cell. This is a diploid cell with one chromosome set, and so there's two chromosomes that are copies of each other. This one came from one parent, the other chromosome came from the cell's other parent. Now when this cell divides by mitosis, it's going to create two daughter cells, one over here, one over here. They're going to end up with exact copies of the chromosomes in the parent cell. So since this parent cell had one pair of chromosomes, both daughter cells have the same pair of chromosomes. They are both identical to the parent cell, and they are genetically identical to one another. So mitosis is going to be the process that happens between the zygote and forming the entire body. So our bodies, for example, are basically made from mitosis after the zygote stage. Now, we're going to look at what happens when mitosis occurs in a haploid cell. This might sound a little bit strange because in animals like mammals, this does not occur. And we'll see that when we look at the animal life cycle, the haploid cells would never go through mitosis. But in plants, haploid cells frequently go through mitosis. So here we have a parent cell. It's haploid, so it has only one chromosome per set, or per pair rather than two. It goes through mitosis, so it makes exact genetic copies. It makes two daughter cells, and each one still ends up with one copy of the chromosome. Again, they are identical to the parent cell. They are identical to one another. So mitosis always results in cells that are genetically identical to the parent cell. Let's contrast that with the process of meiosis. Meiosis is cell division that reduces the chromosome number by half. So daughter cells have half the number of chromosomes of the parent cell. Since there's a reduction by half, only diploid cells can undergo the process of meiosis. A haploid cell already has been reduced, and so it can't go through a further reduction. If haploid cells did try meiosis, they would basically run out of chromosomes. Let's look at the process of meiosis in this diagram. Here we have a parent cell. It's got a pair of chromosomes, and it's going to divide and make 4 daughter cells. Each of these daughter cells is going to end up with half the number of chromosomes as that parent cell. So they each end up with one copy rather than 2 copies. And remember, mitosis gave us 2 daughter cells. Meiosis is going to give us 4 daughter cells. These daughter cells are not going to be genetically identical to each other as they were in meiosis. Some of them are going to get 1 copy of the parental chromosome. The others are going to get the other copy of the parental chromosome. And so this is one of the ways that we get genetic variation among the offspring. So we've talked about how cells can reduce the number of chromosomes through meiosis. The next thing we need to talk about is fertilization. Fertilization is the process wherein chromosomes from 2 cells combine together. The cells that contribute their chromosomes to this combination are called gametes. Gametes are haploid cells that combine in fertilization. In plants as well as animals, the gametes are going to be sperm and egg. The sperm is always the smaller gamete that is more mobile. The egg is always the larger gamete that has more provisions or resources. When they combine together and the chromosomes from each end up in the nucleus together, then that is called fertilization. And as we said earlier, the product of fertilization is a zygote. A zygote is a single cell, and the zygote is always diploid. The gametes are always haploid. So in sexual life cycles, we are going to have the processes of mitosis, meiosis, and fertilization all occur. We are going to have haploid and diploid stages as well. The differences in life cycles relate to whether the diploid cells, the haploid cells, or both go through the process of mitosis. And we are going to use the term generation. A generation is a multicellular stage. It always results when a single cell undergoes mitosis. Let's start with an animal life cycle. This is a simpler life cycle than in plants because there's only one generation. In other words, there is only one multicellular stage. We will also see in the animal life cycle that the haploid cells never go through mitosis, and that will be different in the plant life cycle. So let's start down here at the right with our zygote. Remember, a zygote's a single cell. It is diploid, or it's got 2 N chromosomes. That's a way of saying it has 2 copies of every chromosome. And in this life cycle, the zygote is going to go through the process of mitosis. Remember, mitosis makes genetically identical daughter cells, and so after several rounds of mitosis, we're going to end up with 2 and then 4 and then 8. So this is after 3 rounds of mitosis. We now have 8 daughter cells that are all connected to one another. They are all genetically identical to one another as well. In an animal such as a human, this process would happen many more than 3 times, and we would end up with millions or billions of cells. Now, in our next step in the animal life cycle, at least one of those cells is going to go through the process of meiosis. Remember, meiosis is a reduction division. It's going to reduce the number of chromosomes from 2 of each chromosome type down to just 1 of each chromosome type. And it's going to result in haploid cells. In an animal life cycle, those haploid cells are either going to be sperm or they are going to be eggs. Now, in humans and other mammals, each animal is either male, in which case it makes only sperm, or it's female, in which case it makes only eggs. But for simplicity on this diagram, I have just shown one individual making both sperm and eggs. That's obviously an oversimplification. So we now have our sperm and our eggs. Remember, those are the gametes. They are going to combine together in the process of fertilization and create a zygote. And remember, the zygote was diploid or 2N. That's where the process started. The life cycle can then repeat with mitosis, multicellular organism, meiosis, gametes, sperm and egg, fertilization, etc. So that's a relatively simple life cycle. Only one diploid stage, or only one multicellular stage, and that's the diploid stage. Now we are going to look at the plant life cycle, and you can see just looking at the diagram that this is more complicated. The bottom half of it is actually the same, it's just that we've added a couple of additional steps up near the top. So let's go through it. We will see that there's going to be haploid and diploid multicellular generations, and we are going to see that the process of mitosis occurs not only in the diploid stage as it did for animals, but also in the haploid stage. Let's start down here with the zygote as we did before. The zygote is still diploid or 2N, it still goes through mitosis, it still makes a multicellular stage that is still diploid. There's still some cell or cells that goes through the process of meiosis and creates haploid cells. So that's all the same, except here the haploid cells are not gametes because they are not going to fuse together in the process of fertilization. Instead, we're going to call these haploid cells spores. And spores cannot do meiosis, they cannot do fertilization. The only thing they can do is mitosis. So when they do mitosis, that's going to create a multicellular haploid generation. There's only one copy of each chromosome in each cell. This is something that does not occur in animals, or at least doesn't typically occur in animals that we are used to thinking about. So this entire multicellular organism has only one copy of each chromosome per cell. We need to get to the gamete stage though. So what we need now to happen is for one of these cells, like this one, to go through the process of mitosis. Now in most plants, this haploid individual is either going to be male or it's going to be female. If it's male, then when this cell goes through the process of mitosis, it's going to make sperm. If it's female, then when this cell goes through the process of mitosis, it's going to make an egg. So again, I've simplified the diagram a bit. I could have drawn two of these, one for the male and one for the female, but I kept it simple by just drawing it once. So now we have one individual that's made sperm, another individual that's made eggs. The sperm are mobile, and they are able to be transported or to swim to the egg, and the process of fertilization occurs. After fertilization, we have the formation of a zygote, which is diploid again, and the process repeats. Mitosis, diploid generation, multicellular, meiosis, creation of a spore, one cell, it's haploid, it goes through mitosis, it creates a multicellular haploid generation, and then the process of mitosis occurs, and we get formation of gametes, fertilization, and the process repeats. We are going to introduce two new terms here that apply to plants but are not used for animals. There's no difference to this diagram. I've just added in one of those terms. So we were referring to this individual that was the multicellular haploid generation. We didn't have a name for it before. We're going to call the multicellular haploid stage the gametophyte generation because it makes gametes. So we name the stages in plants after the kind of reproductive body they make. So this is our gametophyte up here. Next, we're going to give a name for the multicellular diploid generation down here. The multicellular diploid generation makes spores, so we are going to call this the sporophyte generation. You can see I added the word sporophyte. The name of the plant life cycle is alternation of generations. It's called this because there's a multicellular gametophyte stage and a multicellular sporophyte stage, and plants constantly rotate between those two stages. The next portion of this lecture will be an introduction to fern life cycles specifically. So please find that video and watch it next.